Hey everyone, so we are going to finish up our practice test EOC review, and hopefully I'm going to try and do the same thing I did last time, except maybe make myself just a little bit faster in going through some of these answers. But we are starting here with question number 28, which was the first number assigned for this last assignment. So 28 is looking at symbiosis as it occurs in roots of some specific types of plants that we call legumes, which is this word up here. Legumes include things like peas and beans and peanuts and chickpeas, and these types of plants are super important when we talk about things like the nitrogen cycle, because their job in working with these symbiotic bacteria is going to be to help add nitrogen into the soil for all of the plants to grow. Remember, nitrogen is going to be important for things like proteins and DNA. And so as living things grow, they need to incorporate nitrogen into those compounds. So we're looking at these root nodules, right, which would contain what we call these nitrogen-fixing bacteria. And those are in this mutualistic relationship with the root of a plant. So the plant would be up here, and then the roots here underground. So the question then says, if root nodules were prevented from forming, which of these would be the most likely impact on the local community? And so if we think about the role of these is to add nitrogen to the soil, well, if we remove that, right, if we can't form these root nodules, then we're not going to be adding much nitrogen to the soil. And so the answer for this one is going to be R. The soil would become depleted of usable nitrogen compounds because we're simply not adding them through this process, right? M doesn't work because photosynthesis has really nothing to do with the use of nitrogen. It doesn't require nitrogen. It doesn't produce it. P, which says the plants would increase water consumption to compensate for the loss of nitrogen. Water is H2O. It has no nitrogen in it. And there's nothing in this narrative here telling me that they would be using water in place of nitrogen. It just doesn't make sense. And then S, the concentration of atmospheric nitrogen gas would decrease, and actually would do the opposite. It would actually increase because there would be fewer nitrogen fixing bacteria removing it from the atmosphere and taking it into the soil. And so, again, answer for this, R, all right? Number 29, which three statements correctly describe the roles of RNA molecules in the process of transcription and translation in animal cells? And so again, you're picking three of these, and those three answers should be B, C, and E. A does not work because it says small RNA molecules in the RNA polymerase. Remember, RNA polymerase is an enzyme that functions in transcription in the nucleus, but RNA polymerase itself doesn't have any small RNA molecule. RNA polymerase also is going to create messenger RNA, but because it doesn't have those small RNAs there, option A here doesn't work. B, messenger RNA molecules carry genetic information from the gene in the nucleus to the ribosome and the cytoplasm, and that is one of those correct answers for this. That's exactly what mRNA is responsible for doing. C, transfer RNA molecules carry the appropriate amino acid to the growing polypeptide. Remember, polypeptide is just the more scientific term for a protein, and transfer RNAs certainly do that process. D doesn't work because it refers to RNA as genomic RNA, which would imply that it's, found in the nucleus, and it's what we harbor and store and pass on to our offspring. And that's exactly what the rest of that statement reads there about transmitting it to offspring. DNA is what's responsible for that, right? Not RNA. So D doesn't work. And then again, E was your third and final answer here with this. Ribosomal RNA function in the assembly of ribosome mRNA complex and the formation of peptide bonds. And that's, again, exactly what ribosomal RNA does. So again, your correct answers for this are B, C, and E. Number 30, scientists sink concrete blocks in oceans to create artificial coral reefs. Which successive event most likely occurs first on this type of reef? So if you think about what makes up a coral reef, we need lots of coral, right? And so you would assume that coral is going to be part of this. When we think about succession, remember there were two different types. There was primary and secondary, and the difference was that primary starting with basically nothing and requires the use of pioneer species to create soil, which then allows plant growth, and then larger and larger things like animals can come in. Secondary differs in that we're starting with whatever was already left over. So you usually have soil available, you usually have some living things already present in that scenario. And so for those reasons, this is an example in this one for number 30 of primary succession, because we're essentially starting with a concrete block and nothing else, right? There's nothing there within which, like, for example, seaweed or giant kelp could start growing. So when we think about the production of a reef, you need to have a couple things. You need to have algae, you need to have some coral, because coral is actually an animal, and coral is going to rely upon something to obtain energy. Coral actually is like a filter feeder. So as like algae and phytoplankton and zooplankton are floating by, they can absorb those nutrients by consuming those organisms. And so actually with this one, usually we tend to think about succession as starting with a producer, but because algae is present in the water, our correct answer for this is going to be P. Coral polyps attach themselves to the surfaces to grow, to kind of create that landing space for more things to start coming in, okay? After all that happens, after those initial stages, then you would get like R here, herbivorous fish, and then carnivorous animals and so on. Okay, but these algal colonies are still going to form, they're still going to be present, but those themselves don't actually physically change the structure of this concrete block. They don't physically allow for a change in a way that allows other things to start growing there, right? They're simply just there and providing energy for the coral. Okay, so again, P is the correct answer for that one. 31 through 34, and actually the ones beyond this are all going to deal with this scenario. If you haven't read through this, pause the video and read through it. That would be helpful. But in general, what we're looking at is the absorption of glucose into a cell. So when something, I think the example, is deer mice in this case, when a consumer consumes food and breaks food down into its monomer pieces, the smaller subunits that make up those large polymers, they're going to start absorbing those molecules. And glucose is one of those super important molecules that we use for energy. The example that they're giving us here is with this transport protein that's embedded in the cell membrane. So here's my cell membrane, or my phospholipid bilayer. And this transport protein is responsible for moving glucose from the outside of the cell to the inside of the cell. So you can see I have a high concentration of glucose here and a lower concentration of glucose here. And this is kind of like a series of steps. Like it first interacts here, it goes through the protein and then to the other side. Okay. So 31 is going to ask, based on the model, which statement best supports the researcher's claim? And the claim is that glucose appears to be moved into cells through facilitated diffusion. Facilitated diffusion is a type of passive transport that does not require energy, just like any other type of passive transport. There is no ATP needed, because molecules are going to move down their concentration gradient. So they go from high concentration to a low concentration, which is exactly the way particles that make up anything, a liquid or a gas, actually want to move. They don't want to be tightly kind of packed together. They want to spread out. So when we think about glucose, it wants to do the same thing, and its highest concentration is outside of these digestive system cells. So it's going to want to move into the cell. The problem with glucose is that it's polar, and it can't get along with those fatty acid tails that we see here in the phospholipid bilayer that makes up the cell membrane. So it does need a transport protein, but that transport protein, as you notice, is not using any energy. Okay. So our best answer for this, given all of that information, is that we're going to move across the membrane A without the requirement for ATP. Okay, if we used ATP, you would see that in the diagram. If we used ATP, there would have been some reference of having to use energy to do this. B doesn't work because it says the opposite. We're going from low to high, and it's actually high to low, right, with diffusion like this. Glucose molecules can't diffuse freely through the membrane without a protein, because you can see here they absolutely need one to get through. And then D, glucose provides energy for protein channel. I mean, that makes sense because glucose does provide energy, but again, none of that is being shown here. All right, so if there was energy used, you would see some indication of that in the diagram. All right, so again, that answer was A. 32. In this case, we're looking at other types of particles that have to move into cells through active transport. And if you remember what that means, if something's active, right, we can think about it using energy, right? If you're super active throughout the day, you're using lots of the energy that your body has stored or that you've consumed in the food that you've eaten. So when we think about active transport, we should see something in these diagrams about energy. We should also see the movement of molecules against a concentration gradient, which means they do not go from high to low, but they go from low to high. So they go from a place where they're pretty evenly spread out to a place where they're going to be really packed together. And again, molecules don't want to do that. So that's part of the reason why we need energy to do this process. M and R don't work because they don't have anything about energy represented, right? Also, M shows going from a high concentration with lots of these little squares to a low concentration with just a few. Same with P, high concentration here, low concentration here. So we're looking for one that goes from low to high, but that also incorporates the use of energy. And so S is your correct answer here. 33, when glucose is broken down, carbon dioxide is produced. You end up with more CO2 inside the cell than outside, and then CO2 diffuses out of the cell. That's the important word there, diffuses. Which statement best describes the movement of CO2 by diffusion? So when we look at this one, again, we're not going to require any energy. We're going to go from high to low. So you're going to want to look for an answer that has something to do with one of those two things. And the one that stands out to me is answer option D. Transport of CO2 through the membrane requires energy from ATP. And we know, we just said, it does not require energy, right? So we can kind of throw that one out. So just the fact that it mentions the energy requirement is not good. Peripheral proteins C help move the carbon dioxide across the membrane. Again, it just said that we're diffusing, right? And we're not going to need the help of a transport or peripheral protein to do this. Additionally, CO2 is not polar. CO2 can easily cross the cell membrane, so it doesn't need any help from those kind of extra proteins. B, water molecules are needed to move CO2 across the membrane. There's nothing in that background information that tells me that, and there's no other molecule that I know of or that you would know of that's going to use water to do that process. And so A is really what we're left with. CO2 can move through the membrane by moving down the concentration gradient, right? From high to low, which is exactly what we think of when we talk about passive transport or diffusion. All right, so 33 is A. 34, which flow chart correctly shows the types of membrane transport? So really, if you see how these are represented, we've got the types up at the top, and then we have energy requirements, right? And so you're trying to pick up the ones that you know you can categorize as either not requiring energy or requiring energy. And of those that we've just talked about, the only one that requires energy is going to be this one here, active transport. So if we scroll down, we want to find one that says active transport under requires energy, and that would be this one right here. Okay, so P is your answer for question 34. If you go down through the rest, you can see where they've made some errors here. This one says active is the only one that does not require energy, so that's flipped, right? It's backwards. And then S, they have diffusion over here on this side under requires energy, and that one just doesn't work there either, okay? So if we go on to part 2 of this, now you look at a food web. In that food web, we're still representing the deer mice that they had been using to talk about the glucose transporter. And now we're going to look at specifically how we convert energy and how it gets passed along a diagram like this. So 35 is asking which pyramid best represents the approximate proportion of energy transferred from the grass to the bobcat. So if I scoop back up, that pathway is going to be from here, grass to leafhopper, deer mouse, to bobcat, right? So I should have 4 trophic levels. The grass, remember, is what we call our primary producer, or just our photoautotroph. Our leafhopper would be our primary consumer, or our herbivore in this case. Our deer mouse would be our second consumer, or what we call our secondary consumer. And then our bobcat, which consumes the mouse, is going to be our tertiary consumer, right? So all of these are heterotrophs here, and then this is my autotroph in the grass. So those arrows, if you remember what they represent, are showing us how energy moves, not how the animals or the organisms move, but how energy gets transferred. So you want to find in these scenarios a pyramid that actually kind of looks in the shape of a pyramid with a bigger base, and it gets more narrow as you go up, because these are supposed to represent the amount of energy, right here, energy available at each trophic level. And so the only one that really takes on that shape is going to be the one provided in D. And it does look kind of odd, right? Because the base is huge and then it gets really super narrow as you go up. But they're trying to kind of show you that D has all of the energy provided in this one kind of sequence of feeding relationships. But as we go up to this, remember, we're losing in these types of diagrams. We estimate we lose like 90% of the energy at each trophic level as we move up to the next. So what we actually transfer is only going to be 10%. All right, and so D works because it's showing that there's really a small amount that gets transferred up, and that's pretty accurate. 36, if the leaf hopper receives 463,000 kilojoules of energy, which value best represents the energy lost between the levels occupied by the snake and the hawk? And this is kind of a complicated way of assessing how much energy is available at these different trophic levels. So if I scoot back up, well, let me think about this. If I scoot back up, I'm going to show you the food web, and then I think how I'm going to show you this answer is going to be, but just kind of holding up my answer key. Okay. So we're looking at this progression right here. We go grass, leafhopper, deer mouse, gray rat snake, and Cooper's hawk. And they're asking specifically about these two levels, the snake and the hawk. So if we start with 463,000 kilojoules here, and we only transfer 10% of that, remember, all I'm doing to that number is moving my decimal over, right? I move it one to the left, every time we go through that energy transfer process. And so if I start with 463,000 kilojoules, let's get this in the right spot, here at my grass level, right? And this is what I'm representing in this position. I get transferred up to the leaf hopper. I'm going to get much less than that, right? I'm going to get 46,300. Well, excuse me. Hold on. I think we started with the leaf hopper, no? Yeah. Okay. Sorry. So I misspoke. So if I start with 463,000 kilojoules here at the leaf hopper level, and then I go up here to the deer mouse, I'm going to remove a zero, move that decimal over one to the left, and I end up with 46,300. If I then go up to the snake, I end up with 4,630. And then if I make my way all the way up here, this is backwards to me, I'm sorry, I get 463 kilojoules there up at the level of the hawk. And so if I want to go in and actually calculate how much energy is actually lost, all I have to do is take the value of the snake and subtract from that the value of the hawk. So essentially it's 4,630 minus 463. And I end up with a number that's going to be very similar in value to one of the answer options, which is P. 4,170. I think it was actually 4,167 or something, but it's going to be close enough in this case. And they also ask for the approximate. So that's how you do that process. So first, again, you have to transfer the energy up and look at exactly how much is there between the snake and the hawk, and then subtract them to figure out what that difference was. Okay. So again, P for number 36. 37, which graph most accurately represents the change in the mass of the DNA in a body cell during one cell cycle. So this is, going back to the cell cycle, one of my favorite things that we talk about all year long because it ties in with so much of what we do in our genetics unit. So when we think about the cell cycle, remember there are 4 stages. There's what we call G1, where cells grow, they do normal functions, and there are checkpoints, right, to check for errors. In S is when we actually copy our DNA. Remember, S stands for synthesis. G2 then comes along, and we do, again, a lot of normal functions and additional growth and checking again for mistakes. And then we have M, which can represent mitosis or meiosis, and then cell division, right? So if I'm thinking about body cells, I'm thinking specifically about mitosis or somatic cells. And in that process, remember, we're going to end up with exactly what we started with. So between my answer options A, B, C, and D, I know I need to find one that I have the same amount of DNA where I started and where I ended. And the only one that really sits there, this one's pretty close. You can see it's a little bit less, and this one's a little bit higher. The only one that ends and starts at the same position is going to be A. All right, and if I check the rest of that diagram, we can make an assumption that G1 is represented here with the kind of normal growth period and the unchanging amount of DNA. And then S by that rising amount, G2 by that stable higher amount of DNA, and then M where it gets divided in half. And you can see again, we return back to that normal amount, all right, at the end of M. So that's exactly what we would expect to see in a cell cycle type of diagram where we're looking specifically at DNA. All right. Now, again, the reason why it works is because we're talking about one specific body cell. Okay. We're not referencing all of the DNA in the body because that would be increasing as we're dividing our cells. But when we think about one in a body cell, we're going to end with exactly what we start with. Okay. So number 38, we have coat color in dogs. It's controlled by a gene that regulates the expression and deposition of pigment. We have patches of color, and those patches in this dog are caused by a somatic mutation. So in other words, a mutation in a body cell. We have different genotypes represented here. And then we've got 3 statements that we've got to figure out which ones would be true of the mutation that caused the darker coloring. So you can see we've got differences in the alleles, right? 2 different genotypes. So it appears as if this is genetic, and it is. And anytime we talk about mutations that are genetic, we know that we're looking at changing a DNA sequence. So in that regard, letter T is going to be your first correct answer because we're looking at, again, a permanent change to DNA in that affected cell. Okay, or in this case, in the tissue that came from and arose from that one mutated cell. If we look at the other answer options, S, the mutation was present in the gametes of the dog's parents, perhaps, but according to the state, that is not a correct answer for this question. And part of the reason for that is because we're looking at what they're referring to as a somatic mutation, meaning it's only in body cells. And the fact that they're using the term mutation gives us the impression that it likely happened after they were born. Okay, otherwise we would just say they had a dominant allele and a recessive allele, right? They inherited the heterozygous genotype, and that's not what they said. So it's not going to be present in the gametes of the dog's parents, so S does not work. R, the mutation occurred in a body cell. We know that because they use the word somatic. And yes, if it wasn't inherited from the parents, it would have had to happen after the embryonic stages, so R is correct. P, also, because this is somatic, it should only affect this dog. So P is also correct. And M just truly doesn't work because we can see there is a phenotype change. All right, so it did affect the phenotype of the dog. To say it was not, you weren't even kind of looking at the picture and considering it. Okay. 39. Graph shows the effect of temperature on the rate of an enzyme catalyzed reaction, right? In the presence of excess substrate. That means we have plenty of reactants that is not limiting to this chemical reaction. So if the reaction temperature were to increase from 40 to 63 degrees Celsius, the reaction rate would likely do what? So if we look at 40, sits here, and this is at the highest point of my rate of reaction. 63 is going to be right about here, almost where it just kind of reaches the very bottom of this diagram. And so if you think about just kind of this portion of the diagram from here over, it's definitely decreasing, right? So we know B and D can't work. If we look at A says decrease because the enzyme would become denatured. Denatured means misshapen, like it's going to be impacted physically. And when it's misshapen, it's saying in this case, it wouldn't function right, right? For C, it says decrease because the enzyme would be used up. And in this case, we're not going to use up enzymes. We never really actually use up enzymes. We simply use them, store them, and then redo the same chemical reactions. If they're changed in some way, right, if they become denatured and structurally they're influenced in some manner that's negative, then that can certainly happen, and then they will need to be replaced. But in this scenario, your best answer is A, decrease, because the enzyme would become denatured. So just remember, denatured means physically changed, okay? All right, number 40, we're looking at a graph that's looking at a change in the size of a bird population over time, and we've got population here and time on the X. And this looks like a really nice little logistic growth curve. If you remember, that is S-shaped, all right? So there is a limit here up at the top, and you can see they've got a question mark there. So the question then is about the space. Which 2 factors can best explain the shape of the area labeled by this? So if we look at our options for this, population immigrated to a different geographical location. If that was true, then the assumption might be that the population size would actually, decrease a little bit. and that's not happening. If you took a whole group of this population and pushed them somewhere else, excuse me, that's going to reduce their population size, right? And that's not happening. P, a treatment for a communicable disease was given to the birds. Possibly, yes, but in that case, it should actually increase their population and size. R, there was competition among the birds for available resources. That would certainly work. That would be something that we call a density-dependent limiting factor. And remember, those control this level up here that we call the carrying capacity. So R is one of the correct answers. S, some birds were unable to find shelter from predators. And if we look at S, that's a potential option as well. That would be a limitation, right? That's another density-dependent limiting factor. The ability to find shelter or housing or the competition for those resources will certainly limit a population's growth. So R and S are correct. T, by the way, does not work because if you introduce a new food source and the birds can consume it, their population is not going to level off. It's going to increase. Okay, so this population size here is very stable. There's nothing negatively happening that's reducing them. It's simply remaining the same due to things like competition. Okay. 41, which two statements support the claim that viruses are non-living? So back in the start of the year, we talked about this concept that viruses, though they have some of the characteristics of living things, they do not share all of them. And so two of them that would point to this idea that viruses are non-living are going to be two that do not fit the characteristics of living things. So we know that living things can evolve and change over time. Viruses also can evolve. So A doesn't work for this, right? Because we're going to try and find and support our claim that they're non-living. So we want to find things, again, not true of living things. Viruses have a genetic code. That is also true. They either have DNA or RNA. All living things are made up of that. And so that one also doesn't work. Viruses lack machinery for reproduction. Well, we know that all living things have to be able to reproduce. And if they can't, then we don't classify them as living. And viruses do not have this machinery. So C is one of our correct answers because viruses themselves cannot reproduce independently. They require the help of a host cell from the individual that they infect. All right, D, viruses contain structures made of proteins. They absolutely do, but so do other living things. And that doesn't really get used often as a factor to distinguish living versus non-living either. E, viruses cannot make their own chemical products, and that is true. And what they're referring to here is like they cannot break down food molecules. They cannot make ATP. They don't use or produce energy in any manner. They just simply kind of float around and rely upon their host cells to do things for them. So your correct answers here are going to be C and E. All right. 42. A segment of a DNA molecule is shown. Which strand is a complementary strand of DNA? So a couple things to point out about this. Remember, this is just a single strand. You can see I don't have a partnering strand. That's what they're asking me about. But I have some things labeled. The P's are my phosphates. These 5-sided molecules with the O up at the top are my sugars. These are my deoxyribose sugars. And they alternate down, right? A's, T's, G's, and C's make up my nitrogen-containing bases, and you can see them lining up at the middle, waiting for their base pairs. But notice the 5 up here and the 3 right here. These are called the 5' and the 3' ends, and it just has to do with what's sticking out at that position. So at the 5' end, we have a phosphate. At the 3' end, we actually have an oxygen and a hydrogen. They're not representing those here. So if I'm looking for the complementary strand of DNA, I almost said RNA. I want to match up my letters here, and I want to make sure my 5s and my 3s are in the right spot. So A pairs with T, and G pairs with C. So my sequence will go something like TAA, CGC, TAA. So if I look for that, TAA, CGC, TAA, and that one looks good to me. But I also have some alternatives here that I need to consider as well. I know U's are not in DNA, so that one won't work. And neither will S for that option. And then this one, they're just not giving us, in this case, the right series to match up because it's exactly the same as what they provided right here. So really my answer is M because that's how they match up. And they do match up in this what we call anti-parallel manner, where one will kind of point upwards and the other is going to point downwards. And that's why we have 3' in this case. That would be up here in the complementary strand, and then you'd have a 5' end down at this end. Okay. All right, 43 through 45, we have another narrative here about crickets, and there's crickets in Hawaii, I believe, and Kauai. And some crickets are identified as having a mutation called flatwing. And in those mutation or those mutant crickets, they cannot make their normal chirping sounds to try and attract a mate, right? They also discovered that there's an invasive species of flies that will actually destroy crickets, and they detect them by chirping. And so the idea here is that if they're not chirping, then maybe the parasitic flies aren't able to find them. All right, so 43, which graph best illustrates the fly and cricket population changes that occurred in the years following the introduction of the flies. So again, the flies are the parasites. The crickets are going to be the host for those parasites. They're going to be negatively affected by them. And so if you imagine a parasite being introduced into this area, well, the parasite's probably going to increase for a little while, right, over a period of time. Meanwhile, your host population is probably going to be all right, and then it's going to dip down as that parasite becomes more impactful on that community. So you want to find a diagram in this case where your fly population increases as your cricket population goes downward. So this one here, A, looks good. This one doesn't fit, right? Because flies and crickets are both going up. This one, crickets are staying the same, and we know that that wouldn't be true if our parasite population is increasing that way. And then here, parasites increases and crickets are little to none almost all the time. So your best answer for this is going to be A, again, for the fact that parasites will decrease the population size of their host, in this case, the crickets. All right, so 44, which statement is the best explanation for the appearance of the flatwing phenotype among male crickets in Kauai? So with this one, we want to consider why this mutation happened and why it's becoming more prevalent, right? And so when we think about DNA mutations, remember, they are not things that are planned in nature, right? They happen at random due to just kind of chance events and errors that happen during DNA replication. And so as part of this, first thing we need to consider is that this is not going to be something that was purposefully added, but that was kind of random in nature. And so if we look at our answer options, there is one that says random in the wording, right? And that is S. So S says a random mutation resulted in the appearance of the flatwing phenotype. So you could have that happen, right? Where you have DNA sequences being randomly changed that then became selected for, right? If we go up and look at R, change in female preference for mates result in the appearance of flatwings. So chances are that that would not be the case because females, again, were being attracted to the sounds of those chirping crickets, right? So they're not going to just change their preference in 10 years or so whenever they did this study. P, infection by parasitic flies caused some crickets to mutate. It certainly wouldn't. And in addition to that, any crickets that were infected by the flies, if you read in the narrative up there, they're certainly going to die, right? The parasitic flies actually consume the bodies from the inside out. So it's not like a cricket can survive that and then pass down its genetic information. All right, so there are a couple things wrong with P. For M, crickets evolved the flatwing phenotype to escape detection by parasitic flies. And yes, that sounds great, except that implies that this evolution process is more purposeful, and it's not, right? If it had said that it randomly this happened and this has allowed them to escape detection, so is being passed on in this group, then that would make sense. But M doesn't read that way. So your best answer with this is S. 45, so they set up an experiment to look at the effects of different variables on fitness. Remember, fitness is a measure of the abilities of an individual to survive better and produce more offspring as compared to others in a given environment. So when we think about this scenario, we've got populations of 50 males and 50 females in 3 different habitats, and you can see some of them have parasitic flies and some do not. All right, so we've got present wild-type male crickets, and we have present female crickets and no parasitic flies. So this is kind of like the original setup, right? Before the flies were introduced on Hawaii. And now here, 2 and 3 are slightly different, right? So which prediction about the cricket population size at the end of the experiment is most likely correct? And so if we don't even really look at those yet, we could say All right, if I don't have any parasitic flies, chances are that population size doesn't change, right? Maybe it even increases if they're really good at attracting mates and reproducing. If I look at experimental habitat 2, where I have wild-type crickets that are actually chirping, I have no flat-winged males that are silent, and I have females and also parasitic flies, chances are that population size will go down because, again, they're being noticed by these parasitic flies. In habitat 3, I have only the silent crickets. I have female crickets, but I have also these parasitic flies. But because the males are silent, chances are that population size is not going to decrease that much. They'll still be able to obtain mates. It's just they may not be able to do so as easily as they could in the past. So the answer for this one is going to be A, population size will be larger in habitat 3 than in habitat 2. It's the only one that works based on those scenarios we just talked about. Okay. 46, part 2 of our cricket scenario here. So now we've got crickets that were on the island of Kauai. Now we have them on Oahu. So we're jumping from here to here. And we're looking at the allele frequencies of this. And this was something that we actually didn't talk about this year with our evolution stuff. And what we're looking at with this, what this means, frequency of flat-wing alleles, the more commonly seen an allele is, the higher this number becomes, all right? And in general, if we say Kauai, frequency of flat-wing alleles, the majority of those crickets found present there are going to have at least one allele of that flat-wing phenotype. Oahu, on the other hand, 2 years later, you can see it's not the majority, right? It's going to be more like maybe half of them. All right, so even though these things are decimals, you can think about them as being whatever's closest to 1 is going to be the most significant, okay? So for 46, which of these observations would provide the best support for a claim that the flat-wing phenotype arose independently on Kauai and Oahu? So again, we want to understand in this case, what is it that would give us the indication that these two phenotypes were not related? They did not come from a similar ancestor. And so if we look at some of these answers, M says the stomach contents of males from both islands contain the same plant material. Maybe they consume similar things, but that doesn't give us any indication as to why these phenotypes are different. Are the same, but appearing in two different populations that are unrelated. So M doesn't really work. P says crickets are transported between the Hawaiian Islands by tourists. If that was the case, then these would not be independently developing, right? These would be things that were actually, there was some mixing between these crickets, moving from one to the other, and it was a shared type of relationship. For R, males from each island have different genetic mutations affecting wing shape. That one would make sense because if there's different mutations, that would imply that there's different communities, right? Which we know we're having because they're on different islands. And then for S, the physical environment and climate on both islands are very similar, and that would only indicate that they should be very much the same. And so with that one, your best answer was R, okay? Because again, there's some differences there, and those differences point to this idea that they came from different places. Which piece of evidence would best help to explain the appearance of different mutations affecting wing structure over such a short period of time? So in other words, how is it possible that this has happened in the order of like a decade when evolution takes such a long time to normally occur? So the first one, populations of crickets experience a high rate of mutation. That's possible, right? In general, the faster something mutates, the more quickly those things can evolve. So viruses are some of the most rapidly evolving non-living things that we have on the planet because it doesn't take much to change the sequence in those molecules. For B, populations of crickets introduce similar mutations to overcome similar selective pressures. Well, that implies, again, in this case, that that's something that's purposeful, and crickets don't introduce these mutations in any way, right? These are totally random. So B does not work. C, wing structure is an acquired trait that's established in every new generation. No, because acquired traits, if you remember, are Lamarck's kind of, it's a keystone of his original theory, which we know was very wrong. The idea that use and disuse is going to either allow you to further develop or get rid of an organ or a structure. Okay, like the giraffes growing the long neck because they have to reach tall trees. That's not what happens in these populations. D, individual crickets determine the wing structure to ensure maximum survival. And certainly kind of like answer option B, that one doesn't work because they simply just don't have that ability to choose, okay? So for 47A is your correct answer. Higher rates of mutation produce many phenotypes and lead to faster evolution. 48, which hypothesis best explains the observed wandering behavior in a population consisting mostly of flat-winged flies? So the scenario was we have flat-winged males that wander about, meaning they move around more than the flies that actually are able to chirp. So the silent flies are moving, the chirping flies, or sorry, the silent crickets are moving, the chirping crickets are staying in one place. All right, and that would make sense if we think about, again, what the purpose of the chirping is. So in the narrative, the original part 1, it was mentioned that these crickets chirp to try and attract a mate. So attracting a mate is going to be kind of compromised if you are a silent cricket. And so moving around is going to be important in that process. So your answer for this one is going to be P. Wandering helps flat-winged males find mates in the absence of chirping. It does not protect them from the parasitic fly. They can't detect them anyway without the chirps. It's simply to attract a mate. All right, 49, rate of cell division. So we've got, I'm assuming, yes, human cells. We have replacement rates represented here and organ systems here on the very, very right. And this says, which conclusion is most consistent with the data in the table? So if we consider these options separately, A says cells in the integumentary system divide faster than any other type of cell in the body. So we can go up here and look. The skin epidermis is integumentary, and that's it. And it takes 10 to 30 days. If you consider, there are some other ones here that are 2 to 4, 2 to 5, 8, 2 to 9, 10. All of those would have been faster because the replacement rate is faster, right? So A does not work for that reason. Cells of the digestive system are among the most rapidly dividing cells in the body. They potentially are. So we've got 2 to 9 days, 10, and 2 to 4. All of those are going to be rapid. For C, cells of the respiratory and circulatory systems have faster replacement rates than those of the digestive system, and that doesn't seem to be the case because circulatory system takes months. And then for D, cells in the reproductive and nervous systems are never replaced. And we can see that reproductive systems are, nervous systems are not even represented, so we cannot make that claim. Your best answer option is going to be B, which makes a lot of sense anyway, because we know our digestive system works hard and works with a lot of acids to try and digest the food that we consume, right? So they need to be replaced because of all that damage that they suffer. 50, which statement best describes an advantage of sexual reproduction over asexual? And there are so many that we can talk about with this. But in general, what we tend to focus on is the variation that we get out of sexual reproduction, meaning offspring are not going to be exactly the same, which allows them to be able to adapt if an environment changes. So M is actually your correct answer right here. It results in more genetic variation in organisms than asexual reproduction does. P, by the way, sexual reproduction does require more time to produce organisms. It's just that is not an advantage, which is what it's asking for, right? That's actually a disadvantage for sexual reproducers. R says sexually produced offspring are more dependent on their parents. That's also true for the most part, not for everything, but for the most part. And again, that's not an advantage, so that one doesn't fit. And then as sexually produced organisms are more dependent on other organisms for reproduction than offspring produced. And that's, again, may also be true, but one more time, is not an advantage, right? So we want to pay attention, again, to what the question is asking for. Okay, ABO blood type. We have 3 alleles. Remember, we've talked about these in the past with our genetics unit. So we got blood type A, blood type B, AB, and then O, which is homozygous recessive, right? It's just little I, little I. So given the pedigree that they're showing you here, they're asking you which 2 possible genotypes could you find for the individual that's got the question mark. So what I did on mine is to write them all out, which is always what I do. Like, I'll go through and I'll write myself a little key about what the genotypes are that I'm looking for, and then I'll actually go through and I'll write them all out. And so on my little pedigree here, I don't know if you can see this, You can see I've got all of the individuals in this family tree represented, or in this pedigree, I should say, represented with their individual genotypes, all right, and what they had to be. So only for those that I knew definitively that had to have that genotype did I write those down. And so notice my question mark person is not written down, okay? But I do know that for that question mark individual there in generation 2, this person right here, their partner here is blood type B, and they came from a pair of parents here that were B and AB. So we know that they had to get a B allele here. We know that they have one child here in generation 3 that does have type B blood, but then we have 2 that have A. So the question mark individual definitely has to have an A allele, because if this parent did, they would have to show it. They would be blood type AB. So we know that they have to have an A, and beyond that, it could be that they are AB because they have, 2 parents and one is A and one is B. Or it could be that they have A and that this parent's heterozygous and they kind of pass down a recessive allele. And so you end up with, these really are your 2 answers for this scenario. You've got B and C are going to be the correct ones. But what's kind of odd about this, and you can see I wrote a question mark there next to B, is the way that they're writing the genotypes. Excuse me. We never write genotypes like this, and this is something that we talked a lot about back when we did our Punnett squares and our pedigrees. Recessive alleles don't get written in front of the dominant alleles, so I'm not sure why they coded it and decided to type it out like this, but B is what the state says is actually a correct answer, as is C. And I would not have a problem with B if it was just written properly, which would be to have that big I, big A, and then the little I to the right of that. And they just kind of flip-flopped them. So just be aware of that and think through, okay, which ones are definitely not options? We know like A was not an option here because you had to have an A allele coming from somewhere. And same for these 2, right? You had to have an A for that other parent with a question mark. All right, so for 52, role of plants in the cycling of matter. And so we're going to grow plants under conditions that limit the rate of photosynthesis. So in other words, what are going to be some limitations to that? Remember, photosynthesis, in terms of its reactants, is going to require the use of sunlight, it's going to require the use of water, and it's going to require the use of carbon dioxide. So if we look at M, grow the plants in a low oxygen environment, that does not work because sure, it may limit the rate, but plants don't even use oxygen in that case, right? We're not measuring CO2 in this process, so it doesn't work for the purpose of the study. P, grow the plants in a low CO2 environment and measure the rate of oxygen. That one seems to be good because plants do produce oxygen through the process of photosynthesis. R, grow the plants in soil containing excess, meaning additional or extra water. Measure the rate of transpiration. That is not photosynthesis. That's water loss from plant leaves. And if we have excess water, that's not limiting anything. If anything, it would be speeding up photosynthesis. And then S, grow the plants in soil containing excess nitrogen. That's all well and good, but nitrogen, again, doesn't have anything to do with this photosynthesis process. And so your answer for this one is going to be P, okay? 53, dialysis, and this is what we call a dialysis bag. It's used to kind of represent osmosis and the movement of solutes or water molecules really across a membrane. So here's kind of what we need to know about this scenario right here. So it says students set up an investigation with a bag containing 2% iodine solution in a beaker that has 9% starch, and they're labeling this here for you. This statement right here is really important. So starch turns blue-black in the presence of iodine. So meaning if iodine leaves this bag, or if starch gets into this little bag, that's where we're going to see some of that blue-black color coming through. The dialysis bag is permeable to water, meaning it lets water in and out, and iodine, so it lets iodine also in and out, but it cannot let starch in and out. And that makes sense because starch is a very big molecule. It's a polysaccharide. So from that, we want to pick 2 observations that accurately predict what will occur in the investigation. And so in this example again, if the dialysis bag can move water and iodine, we're going to get some movement of something here. So I only have got 9% starch on the outside. It doesn't seem that I have any starch in here. And so going back to what I remember from osmosis, I would call my solution a hypertonic solution because I have a lot of starch, a lot of solute present outside the dialysis bag, okay? Now the dialysis bag not having any is what we call hypotonic. And if you remember from osmosis, what we were talking about is that water always goes to whatever's hyper. So water is going to leave the dialysis bag, all right? And as a result of that, my starch solution is going to increase in terms of its volume. But it's asking also about the presence of iodine. And I don't start with any iodine outside here, which means there is a concentration difference, right? I have iodine inside the dialysis bag, but I don't have any out here. And so iodine will also diffuse out of the dialysis bag and into the surrounding solution, which in turn will turn that starch blue-black. So my answers for this one, the two of them that are going to work, are going to be B and D. So the solution turns blue-black due to iodine leaving that dialysis bag. The mass of the dialysis bag decreases as iodine and water both leave. All right. And even the volume of the solution would also increase, but I don't have that as an answer option. Okay. But the solution dialysis bag does not turn blue-black because starch can't get into it. Concentration of starch in the bag will not increase because there's none there to begin with. And again, starch can't move across that membrane. And then the mass of the dialysis bag will not increase because there's nothing going in. Both iodine and water are both going out. Okay. 54, the very last question on this. The graph shows the results of an investigation into the effective distance from a light source on the rate of photosynthesis. So again, the further away from light source, the lower the rate. The closer you get to a light source, the higher the rate becomes. So based on the data, and we're kind of filling in these blanks here, the rate of photosynthesis does something as the level of available something else increases. The greatest change in the rate occurs when the light level is something else. So if we look at our options here, M says, and if we stick those words in these positions, based on the data represented, the rate of photosynthesis increases, which it does, as the level of available light increases. And that makes sense, right? Because we're going up here as the amount of light is also increasing as we get closer to our light source. The greatest change in the rate of photosynthesis occurs when light level is low. And that's also true. We don't get a ton of changes up here. But when we're very far away, we see this steep increase because there's a significant difference in the amount of photosynthesis that can be done at a distance. So M looks good. And if we look at some of the other ones, you can see some of the other options about oxygen, oxygen again. And then this is actually S is really the opposite of what we had for M, at least in terms of this first in number 1. So of those answer options, M is the correct one. And again, it's because those words fit in those blanks accurately. Okay. So finally, that's all I've got here for the last of this practice test. If you need anything or have any questions for me about these items, let me know. I will be posting an answer key for you on Canvas with just, the bubbled in answers as well. So if you want to just go that route, by all means, you're welcome to do that. But otherwise, I will see you on a Zoom call or in class, and we can talk more after EOC. All right. If I don't get to talk to you until then, good luck on that EOC on Tuesday, and I'll see you sometime soon. Bye, guys.